For the loop of Henle, it is the structure that is present after the proximal convoluted tubule till the distal convoluted tubule. You have thick descending port, thin ascending port, thick ascending and thin ascending. Here, as you can see, these are the port where the distal convoluted tubule is present. This is the thin port, okay, and this is the collecting tubule. All of these structures are present in the medulla. Its function is to produce hypertonic urine and maintain and protect the body water by reabsorption. The distal convoluted tubule is shorter than the proximal, and it is formed of three parts: straight part and convoluted part, and then the macula densa part. This is the distal convoluted tubule. It is formed of 6 to 10 cells. The cytoplasm is acidophilic but less acidophilic than the proximal. This is the proximal convoluted tubule. And the lumen of the proximal convoluted tubule is filled with microvilli, whereas the lumen of the distal convoluted tubule is free of microvilli, because the microvilli in the distal convoluted tubule are very short compared to the proximal. This is the distal convoluted tubule. The microvilli are very short. You have a lot of mitochondria for ion transportation, and you have little pinocytotic vesicles at the surface. Okay. Its main function is the reabsorption of water, sodium, and the excretion of some sodium and potassium. The collecting tubule is not a part of the nephron. There are three regions of the collecting tubule: cortical, medullary, and papillary. The papillary are the end, the final stage, which open into the papillary renal papilla. Okay, so here, this is the cortical part. This is the medullary part of the collecting tubule. This is the papillary duct that would open into the papilla. Okay, this is the collecting tubule. You have pale cells and you have dark cells, and its function differ. The dark cell contain lots of mitochondria. It is for collection of urine and concentration of urine, regulated by secretion of potassium from the principal cell and reabsorption of potassium by the intercalated cells under the control of aldosterone. It would maintain the acid-base balance of the body and the urine. So again, the nephron you have here, this is the glomerular capsule or the Bowman's capsule, contains the glomerulus, proximal convoluted tubule, have many microvilli, whereas the distal convoluted tubule less microvilli. The loop of Henle is formed of two parts: descending part and ascending part, thin and thick part. The thick part is lined by cuboidal epithelium, and the thin part is lined by simple squamous epithelium. You have cortical collecting tubule and medullary collecting tubule and papillary collecting tubule. You have pale and dark cells, which are principal and intercalated cells of the collecting tubules. For the juxtaglomerular apparatus, it is formed of macula densa, which is part of the distal convoluted tubule. You have juxtaglomerular cells, which is part of the afferent arteriole, and and you have flaccid cells, which is the extraglomerular mesangial cells. Okay, this is the macula densa. Okay, the cell appear columnar, and this is the juxtaglomerular cells. They are part of the afferent arteriole, which is the entry of the blood vessels, and this is the last cell or extraglomerular mesangial cell. Okay, these three structures form the juxtaglomerular apparatus. It faces the glomerulus. This is the macula densa. Macula densa again. The macula densa senses the presence, increase or decrease in chloride ions in the urine, and it sends its feedback to the juxtaglomerular apparatus, and also sympathetic nervous system would send feedback into the juxtaglomerular apparatus, juxtaglomerular cells, and they would secrete renin. This is the macula densa cell. And these are the distal convoluted tubule cells. As you can see, the macula densa, the cells are much longer than the distal convoluted tubules. The distal convoluted tubules, the cells are cuboidal in shape. Their microvilli are short, whereas the microvilli of the macula densa cells are long. You have lots of processes of the macula densa so that it could reach the juxtaglomerular cell. The juxtaglomerular cell contains lots of electron dense material present inside it, which is the renin hormone present inside it. The macula densa main function is to detect, monitor the chloride ions and detect the flow rate on the tubules. It is sensory. Juxtaglomerular cells are modified smooth muscles in the afferent arteriole. This is the juxtaglomerular cell. Its function is to secrete renin. The renin would change angiotensin into angiotensin 1, and then angiotensin 1 would change into angiotensin 2. That would allow the release of aldosterone, okay, to increase the blood pressure. Structure function adaptation. The cells of the macula densa is close to the juxtaglomerular cells. Absence of internal elastic lamina in the afferent arteriole, and presence of sympathetic nerve fibers that supply the juxtaglomerular cell. The function of the renin by different ways, two main ways, by angiotensinogen and by aldosterone. For the last cells, they are extraglomerular mesangial cells. The renal interstitium, you have the interstitium is just connective tissue. It is any part where the distal convoluted tubule or the proximal convoluted tubule come together like this. These are the whatever distal loop of hand whatsoever. The space in between, this is the renal interstitium. This space in between, this is the renal interstitium, and it is filled with connective tissue. Okay, like any other structure. The connective tissue, of course, contain fibroblasts and macrophage. Okay, in the cortex, it is fine and minimal because of the presence of lots of tubules in the cortex. Okay. In the medulla, you have medullary interstitial cell, which is the medullopine, which would secrete medullopine 1, which decrease blood pressure. So the kidney have dual effect. They have medullopine. The medullopine decrease blood pressure, and renin increase the blood pressure. The structure of the ureter, as we have said in the beginning of this lecture, it is epithelium mucosa, formed of transitional epithelium and lamina propria. Lamina propria are just some loose connective tissue present. The transitional epithelium appear to have facet cells on the apex. Okay, facet cells. And you have muscle layer and adventitia. For the urinary bladder, the transitional epithelium a little bit thicker This is the lamina propria, and this is the muscle layer. The muscle layer is much thicker than in the ureter. And then you have the adventitium, the last part. The lumen of the urinary bladder is wider and folded. Again, this is the transitional epithelium, lamina propria, smooth muscle fibers. And then this is the transitional epithelium again. The transitional epithelium is usually formed of 4 to 5 rods of cells. The apical cells are binucleated or mononucleated cells. Okay. The cell membrane of the facet cells or umbrella cells, the cell membrane that faces the urine is thick to avoid any urine material to affect the cell membrane, and you have reserve of the cell membrane present in the cytoplasm that would join the surface to increase the surface area. Here again, the cell membrane on the apex is much thicker than any cell membrane any other place, and this is a relaxed state, so it have lots of cells, whereas in the stretched state the cell would increase its length. For the male urethra is about 15 to 20 centimeters. The bladder is lined by transitional epithelium. The prostatic urethra is also lined by transitional epithelium. The membranous urethra is lined by stratified columnar epithelium. And the penile urethra is lined by stratified columnar, but the last area, the fossa navicularis, is lined by stratified squamous non-keratinized epithelium. Okay, the prostatic urethra is about 4 cm in length, and it is present inside the prostate. It have a V shape, whereas the membranous urethra is about 2 cm, and the penile urethra is about 12 cm in length. Okay, I hope the best for you all, and that you memorize everything in the histology lectures, and these are the references. Thanks and